Now, this is something that I do regularly. So I have the protocol for you. First thing that you need to understand. Now, here I'm going to just show you the steps. I have divided it into 7 steps. Basically, you can actually go more than this, but in general, we can say there are 7 steps. The first thing is retrieving the molecules. That something that after you decide which molecules, which protein and ligand you are going to work with, you have to retrieve them from specific, uh, sources, websites. We'll come to that. And then second thing, after you have retrieved, you have to prepare for the protein molecule, prepare the protein molecule for docking, and then prepare the ligand molecule for docking. Both of these things done, you have to prepare the grid parameter file, which we had said there is a space that we are going to fix. So this is the three-dimensional space where search is going to happen. So it's called the grid parameter file, GPF file, in Autodoc. Okay. So, or otherwise it's also called grid. You can simply call it as a grid. And then we go for creating doc parameter file, which is the extension of which will be DPF. And doc parameter file decides what kind of docking procedure you are going to adopt, which algorithm you are going to adopt, how many times iterations will be there, things like that. Okay. And after everything is set, we are going to run auto grid and auto dock. So that is where docking happens. And finally, once you have the docking done, comes the next bigger section, which is visualization and analysis of the dock molecule. There are so many software available for this purpose, and even online there are tools available. You might have heard about Rasmol, Pymol, Chimera, so many. There are many more. So you can use whatever you want, anything that you like. Over here, we are not going to explore multiple things. I'm going to explore, show you how we are going to use Chimera. After I have an experience of almost 10, 12 years on doing this docking thing, and I figured out Chimera to be one of the best tools, not the most user friendly, but once you learn it, it's a really good tool. And yeah, so we will be using that. Now, see, the images that I have shown here does not represent everything that is mentioned over here. Only representative images are there. So what are the tools for docking? These are the minimum things that you require for docking. Okay, the first thing is Autodoc 4.2 and Autodoc Grid 4.2. That is obviously very essential. Then you have MGL tools, which is also called the Autodoc tools. Okay, so this is the web page. This is how the web page looks now. You can go there and you can download the software pertaining related to your specific system, and then you have Chimera or Pymol that can be used. So I've just shown how a Chimera page, when you open it up, open Babel. This is something that we will be using. Yes, here we are starting. I'm going to tell you the steps. I'm just telling you the steps right now, and then we are going to do it. Okay. So first learn the steps, and then when we do it, when I'm going to show how it is done, you can relate to this. So we are. This is the first step: retrieving the molecule, proteins and ligands. So once you have decided which protein, this is the website rcsb.org, which is also called the PDB, Protein Data Bank. So how Protein Data Bank looks like, and you have to go there. You have to search for your protein, and then you have to download that. Okay. So usually when we are trying to do docking, we should see that the protein that of our interest is not mutated at any place. So no mutation. You should see that. Now once we go to PDB website, I'm going to show it. Then what do we do? How these are edit and save PDB text after removing dimers and retrodimers for ligands that you can get from the PubChem website. Okay, the molecules, uh, the ligand molecules are retrieved from PubChem, and we download the 3D SDF format of that SDF. There will be so many formats. We'll see that also. So 3D SDF format that will be downloaded from PubChem, and then we convert PubChem. Sorry, not PubChem. We convert the 3D SDF format of the ligand molecule which is downloaded in PDBQT format. This is uh, step three from the here from the protocol that I've shown you, preparation of ligand. So there is another way of preparing ligands, but this is a very quick method, and I have tested it. It works perfectly fine. So you can go with this. It's not the only method. You can. There is another lengthy process, but yeah, you can go with the shortcut. So this is open label tool. How it looks like. So in here you have a left hand side and you have a right hand side. Uh, in the center you have some parameters that can be fixed, and on the left hand side you can choose which uh, particular ligand you are going to format. You have to first select the format here, input format, and here is the output format. So you, after setting up the input format to SDF, you choose your ligand molecule, and then you fix PDBQT as the output format, and the ligand output over here will come. You can just save the name and press convert. It will get converted to that particular format. That is the only purpose why we use open label, nothing else. After this part is done, we go to the Autodoc tools, okay, where we are going to prepare the protein molecule. So there are these four steps that are required when you are going to, convert the protein molecule in the particular in a specific format, that is PDBQT format. See, Q and T is charge and torsion. Q is for charge and T is for torsion. So these are the two driving forces for docking, and that's why we call them PDBQT. What we do is the first step after you have the protein. Usually we take the monomer. We don't take the dimer form of the protein, even if the form that you have downloaded from PDB is in a dimeric form. You will remove the one of the monomer, okay, or if it's a trimer or tetramer, you remove all the other monomers. You just keep one, okay, and then you also remove the extra ligands that will be there already. When you download it from the crystal structure, you will have some ligands already there. Usually they are inhibitor molecules. So we take them off, and then let this space be free. And then what we do is we open them in Autodoc tools, and then we add some polar hydrogens into them, and then we add charges into them. Coleman charges are the most preferred charges. So we add Coleman charges into them, and we assign them in a specific format, AD4 type, which is Autodoc 4 type of atoms. So the atoms are assigned AD4 types. Then we save them as PDBQT. Simple. Just three steps, and fourth is saving. So that way you're going to create your PDBQT file of the protein. Now, next step, you already have the you have downloaded the molecule, you have the proteins which you require in the PDBQT format, and you have the ligand by this time in the PDBQT format. So next is creating the grid parameter file and then the doc parameter file. So what we do here, we create a grid. Grid is a space where docking will happen. For that, you need the macromolecule in the PDBQT format, and you will have the ligand, which can be opened from either the molecule, or you can also go to set map types and open ligand. In the PDBQT format, you open both of these things, and then we choose the. After you open, it doesn't mean that you have chosen. So you have to choose both the protein as well as the ligand. So you choose the protein, and then you choose the ligand, and then you create the grid box, which is usually uh, we start with the center atom of the macromolecule. So once you this is the is the box which will pop up when you are trying to make the grid. So this is the grid box. It's a three-dimensional cuboid which whose sizes can be fixed, and I mean there are certain things that you need to take care over here, which when we are doing the docking. Okay, finally once you have done that, you are going to save the options, the grid options that you have selected, and then you save the GPF file. So this GPF file is going to be the one where which we are going to use AutoGrid tool, and then we are going to create the grid. This is just setting up the parameters for grid. Okay, grid parameter file. Now we have to create the grid. Now after that is done, we create the docking parameters. What will be the parameters for docking? For that you need the macromolecule. So if it is not open yet, because since you have opened it in the last step, it will be still there. So you can just go with the set rigid file name, which is for the rigid flexible docking. See, there are two options that will pop up when you are doing this. There is a set rigid file name and there is a flexible format also. So in here, in today's workshop, we are focusing only on rigid flexible docking, and this is the one that is usually used almost everywhere. Okay, unless you want to make the protein flexible also. If you try to make the protein flexible, you have to have a very good uh, processor which can handle that much of because otherwise if both protein as well as the ligand are flexible in nature, it's going to be difficult for any normal system to handle that. So, and that's not necessary also. What we try to do usually is we assume that the proteins will not be flexible, will not be opening up or closing, or the amino acids will be moving around. Unlike something that you have studied in your probably in your enzymology classes, that enzyme will open up and the ligand will, the substrate will go inside, something like that. So that we do not assume all those things. We just assume a space using the grid, and then we go for docking. I mean, setting them rigid, and the molecule we are keeping at, I mean, the ligand we are keeping flexible, which can move wherever movement is allowed. So set PDBQT. We have selected the macromolecule, the protein molecule. Next step, we have to select the ligand. So we choose the ligand, and then we have the search parameters where we choose genetic algorithm, one of the most trusted algorithms for nowadays for almost everything, not just docking. So genetic algorithm, we can set up a run of 10 to 50 depending upon what exactly you're going to do. And I mean, you can go more than 50, obviously, there's no problem with that. The more runs you are going to do, the more time it is going to consume. Usually, when there is a lot of molecules in the protein, very dense area, and you have a large molecule ligand which has to be searched, we go for higher number of runs, like 40 or 50, or depends. But otherwise, a run of 20, 25 is good enough. But we are not going to do 20 runs today. 20 runs takes time. So just to understand how things work, we'll keep it short. We'll keep it at 10, or if you want, if time is not really sufficient, we'll keep it even 5, so that you can understand how things are working. So we set up the number of genetic algorithm runs, the genetic algorithm parameter, and finally we save. We choose the output as Lamarckian genetic algorithm. There are two options of genetic algorithms over there. So we choose Lamarckian and save as the .dpf. This is the dpf doc parameter file, and next up you have the grid parameter file. So you, by this time, you will have all the files necessary for docking. You are ready for docking. So how do we do docking? We actually go to the command prompt. And this thing you will have to learn, because you have to go to the specific directory where you have saved the protein molecule and the ligand molecule. So before we do anything, One thing that I have not told you, but definitely when once we start, I'm going to focus that on again. You have to have a folder, okay, at a specific location, and you are going to keep everything in that. Otherwise, it is going to be a mess. So for we wherever we have saved all these files, routines and ligands and grid and doc files, we have to choose go to that directory using the command prompt. And there we are going to save the autodoc 4 and autogrid 4 exe files. Okay? And once you have it there, these are the two commands that you will be requiring. You may also make a note of these. I should rename this. This is autogrid 4.exe dash p grid.gpf. Instead of that, I should have written file name .gpf and dash l log underscore gpf .glg. Instead of that, I should write file name over here. So I just correct this. So we are here. This is the command that you are going to use for auto grid. So please make a note of this for a quick reference. You're going to write in double quotes. You will write autogrid 4.exe dash p, that is parameter. This is for parameter. This is denoting parameter file. So the name, whatever name you have kept for the GPF file, you're going to write here .gpf dash l, which is creating a log of the same thing, whatever is going to happen. So dash L file name dot gl single quote. No, that has not to be included. Still, I'm not going to remove it now. We'll do it later. After that completes successfully, we are going to run autodoc with the following command, which is very similar to autogrid. If you don't have autogrid here, you have autodoc instead. Autodoc 4.exe dash p, same thing. File name dot dpf over here because you are running doc parameter file. So the parameter file name is given, and then dash L where log will be created, file name .dlg. That's simple. Talking will begin, and you will have a log file in the destination directory with the name of file name .dlg. You have to wait for the completion, which will depend on the number of runs, as we have just told. And just to show an example, uh, this is the command prompt, and we have gone to the this is docking.demo. Docking demo is the folder where everything is kept, and this is the grid running grid command autogrid.exe dash p. This is the file name I have kept. So stepbuyer.gpf dash l. So there's a space over here. Just have a look very clearly where you have to keep the space and where you are not supposed to add a space, because in command prompt, errors are very often. And if you get errors, you will not be able to complete it successfully. So yeah, dash p stepbuyer.gpf dash l stepbuyer.glg. Once that is done, you will get a command line, something like that. In that, you're going to type autodoc 4.exe, and the same thing that I just told you. And once that is done, again it will come back to this. So once the docking is done, after successful completion that you will see here, with this line coming up back again, you you will understand that docking is done. You can also check. We'll also have to check the size of the file that has been created, the DLG file that has been created. Finally, once that is done, we move on to visualization. So visualization, we are going to use Chimera over here. It is where you learn how, where the molecule has docked. So the first step is opening up the DLG file, which you will see, which you have to go to that particular library directory. Okay, sorry. You have to go to the directory, and then you have to open it in PDB format. Once you open that in the PDB format, this is the docked result. So there are three poses that has come up after running the autodoc for streptavidin biotin, and they're all three at the same location but in different orientations. They look very jumbled up. Now, in here we are trying to just see. Look, all the three are there. Not I have not removed anything from there. And there is this protein structure hidden. This is biotin molecule and this is structure hidden. And this is the location where it is binding. What are the amino acids which are making a hydrogen bond with this? All those stuff we are going to see when we are going to do docking.